Buon pomeriggio a tutti, bentornati. Io sono Simone Del Favero, Università di Padova, Dipartimento di Ingegneria dell'Informazione, ed è veramente un piacere introdurre questa sessione in cui verremo condotti nell'affascinante mondo della biologia sintetica e della bioinformatica. Lascio immediatamente la parola alle chairpersons di questa sessione, Lorenzo Pasotti, Università di Pavia, e Linda Pattini, Politecnico di Milano.
Benissimo. Molte grazie, buon pomeriggio a tutti, benvenuti a questa seconda parte della Benvenuti a questa seconda parte della prima sessione. Questa seconda parte è dedicata alla bioinformatica e alla biologia sintetica. Come detto, abbiamo 3 contributi in particolare. Ricordo agli speaker che hanno 10 minuti, i contributi si succederanno uno dopo l'altro e chiuderemo con la sessione delle domande alla fine. Quindi adesso prego il primo speaker di venire al podio. Abbiamo Deborah Dall'Era con la presentazione Study and Characterization of Bile Acid Biosensors in Engineered Probiotic Bacteria. Please.
Thank you for the introduction. Today I'm talking about synthetic biology work on study and characterization of bile acids biosensor in engineered probiotic bacteria. So we will talk about the bile acids, their physiology and their relevant, clinical relevant applications, biosensors in synthetic biology, and then which are the main focus of this work, the results that we obtain after the characterization of bile acids biosensor and 2 different applications of these biosensors. So first of all, we are talking about bile acids. These are molecules, amphipatic molecules produced during the cholesterol pathway as primary bile acids. They are immediately conjugated with glycine or taurine, and they are secreted in the intestine by bile. Here, the gut microbiota operates a list of transformation over them, such as the deconjugation by the bile salts hydrolases, the BSH enzymes, and then the transformation in secondary bile acids. 19% of the total bile acids pool bile acids, is reabsorbed by the enterohepatic circulation, while the 5% is excreted by feces. These molecules are involved in some important metabolic pathway, and they can also control the overgrowth of pathogens such as Clostridium difficile infections. In fact, some of these molecules, such as the primary and conjugated bile acids, can enhance the formation of this infection, while the secondary and unconjugated bile acids can prevent these infections. Normally, these molecules are measured by mass spectrometry, but in this work we use biosensors. So what are bacterial biosensors? Bacterial biosensors are cells containing a genetic program that enable them to sense external input and respond with a specific output. The regulated promoters contains an operator site that recognizes a transcriptional factor that can be an actuator or a repressor, and the presence in the on state, the presence of both input and the transcriptional factor enables the transcription of the gene of interest, so we obtain a measurable output. In the off state, the absence of the input inhibits the transcription of the gene of interest, and so we obtained no output. This biosensor can sense different type of inputs and also can respond with different type of outputs, and then they can be combined to create complex genetics circuits. The aim of this work is to evaluate bile acids biosensing circuits in E. coli for therapeutic applications to detect and regulate specific classes of bile acids in different case studies. So first of all, we have to characterize bile acids biosensor in bacterial strain. Then we can apply this biosensor to characterize transforming enzymes and to create a regulator, closed-loop regulator, to control the concentration of these bile acids. First of all, we, as I said a few moments ago, we characterize bile acids biosensors. So we characterize 4 bile acids biosensors. They are all repressor. So in presence of bile acids, the transcription is works. We tested all these biosensors in 2 different strains, E. coli top 10, which is a laboratory strain, and E. coli Nissol, which is a probiotic strain already used as a drug. The results in these 2 strains were comparable, and we tested this biosensor with a list of bile acids, and we measured the absorbance and fluorescence using a microplate reader. At the result, we obtained that these two biosensors doesn't work very well. So we focused in the first 2 biosensors. Here we can see the results of the RFP per cell of TCPPH bacterial biosensor and VTRAC both in E. coli Nissol. And what we can see is that these 2 biosensors seems to be specific for a group of bile acids rather than a single bile acid. So TCPPH seems to be more prefer the conjugated bile acids, while VTRAC seems to prefer the secondary conjugated bile acids. And here there is an example of fitting of experimental data of the biosensors TCPPH with bile acids. So getting these results, TCPPH seems to be the better candidate to be used to make a sort of screening of the enzymes BASH that, as I said at the beginning of this presentation, is the enzymes that deconjugate the conjugated bile acids. So we can use expressing vector expressing different genes, BASH, and under the control of IPTG with different concentration of IPTG. And if we test these expressing vector with different bile acids, conjugated bile acids, we obtain at the end of the experiment samples that contain an unknown amount of conjugated bile acids that has not been transformed by the enzymes. So if we want to retrieve the amount of conjugated bile acids that has not been transformed by the enzyme, we can use this sample to induce the TCPPH enzyme biosensor, and so measure the steady state fluorescence with these samples, and then compare this value with a calibration curve of the biosensor. So here we can see an example of what happened when we use the BASH-B from Lactococcus johnsoni with 3 different bile acids. In the first case, with the TCA bile acids, we can see that at time 0, the concentration of bile acids is higher rather than in the time 24 at the end of the experiment. While, for example, in the third case with the TDCA, the level at the beginning and at the end of the experiment are very similar. So in this case, the enzymes seems not to work. So the best performing ones were selected for dynamic characterization. We sampled the culture at a specific sampling time in order to study the dynamic of the deconjugation. And here we can see an example of fitting of the experimental data using this equation, where the constant Ka is the rate of the conjugation of specific bile salts hydrolases with a specific bile acids. Okay, so these two biosensor and enzymes can be put together in the same bacterial cell in order to create a closed-loop regulator of bile acids. So first of all, we have to design the mathematical model that represent the activity of this system. The first equation represents the growth of the cell. The second equation represents the controlled production of the enzymes. And the last two equations represent the formation of the transformation of the substrate. So the production of the BSH is controlled by the enzyme, by the biosensor. In fact, we modeled it with a nil function containing the constant value of that we obtain during the characterization of the biosensor. The biosensor. In the last two equations, we can see that there is modeled the activity of the enzymes using the Ka constant that is obtained during the characterization of the activity of the BSH. So in the first plot, we can see that the conversion of the conjugated bile acids is controlled by this closed loop regulator. In fact, the steady state value is reached under 2000 minutes, and this value corresponds to the Km of the biosensor. And it seems to be robust, varying the OD of the culture. In the second plot, we can see that the BSH is produced in a controlled way by the cells. In fact, at higher OD, we can see that is needed a lower quantity of enzymes rather than when the OD is lower. So let's summarize the main point. We demonstrated that the potential utility of the bile acids biosensor for clinically relevant proof of concept application. Biosensor can be used in probiotic to interface any complex logic function for in vivo application. Each application we have seen needs optimization to expand the collection of bile salts hydrolysis and implement the regulator for future in vivo application. So let me thank all the components of the lab, and also thank you for your attention.
Thank you, Deborah.
Thank you. So we move to the next speaker, who is Massimo Bellato from the University of Padova, and he will present a work dealing with modeling metabolic overload effects in bacterial growth rate in synthetic biology. So the stage is yours.
Thank you for the introduction. So let me just steal a minute before starting, because I want to share a fun fact that I realized yesterday. So I've seen that, looking at the date, today is exactly 7 years since my first oral presentation in a conference, and it was actually at the 5th Congress of the GMB back in Naples. And I was working as a first-year PhD student in the same laboratory of the previous speaker. So I'm particularly happy to be here today and actually being able to talk after Deborah. Now instead, I work at the University of Padova in the Department of Information Engineering, and I collaborate with the Molecular Medicine Department in the Synthetic and Systems Biology team. And for the sake of honesty, the work that I'm presenting today was almost everything done by this great master student that I had supervised last year. That she wanted to start dealing with the biological systems. Probably I haven't done a perfect job with her, since now she's working on autonomous systems in another university, but she promised that she will be back in our community, so hopefully, let's see. So Deborah already said that synthetic biology means trying to place information in the form of genetic circuits inside a cell, so that those cells are then able to get an external stimuli and produce an output that can be either the production of a protein or a certain behavior and so far and so forth. And this essentially comes from the fact that those genetic circuits are DNA that are transcribed into mRNA. Those mRNA then is translated into proteins by the ribosomes. But the thing that is maybe less known is that those ribosomes actually are a limited resource inside the cells. Indeed, it has been proved that, for example, if you place in a cell two expression cassette, one constant for a green fluorescent protein, a GFP, and one inducible for a red fluorescent protein, an RFP, both very strong, the higher is the induction of the RFP, the weaker will be the signal given by the GFP, and this leads to the sharing of these translational reserves. Typically in synthetic biology, you try to deal with it by modeling, like saying, well, I know that I have a certain amount of ribosomes, they're going to be split among the different mRNAs, and you try to design as best as you can your circuit. However, in literature, there is another aspect that is related to the ribosomes, which is how ribosomes are affecting somehow the growth rate, which is how fast a cell grows. And especially it has been shown that linear dependence exists between the number of ribosomes and the growth rate of the cell. Even if those studies has been done mostly by touching an endogenous system of the bacteria, which is the SPO-TRLA system, that is more related to the nutrients availability for the cells. So essentially what happens is that typically in synthetic biology, when you design your circuit, you try to deal with the metabolic burden, and if you see that your circuit is demanding too much and you have growth rate defect, you just say, well, I've done a bad design, go back from scratch and start again in the design. However, nowadays, we're trying to move a step beyond the design of a single strand doing something, but trying to engineer communities of bacteria. And in this case, sometimes it's actually something that you want to be able to tune the growth rate and also maybe change the general behavior or the phenotype of the bacteria that compose your community. So we try to understand, well, how can we mix all those models, like trying to take into account both of the effects, considering that, for example, the models that involve the ribosome related to the growth rate typically says that once you decrease the ribosomes, you have a decrease in growth rate, while we have observed that exists a range in which you apply load to your cell, but nothing happens to the growth rate. Moreover, it's very difficult to have a measure of the ribosomes which are binded to which type of mRNA. And so essentially, we tried to first take some of the most used architectures, let's say genetic architectures describing this type of system, and trying to nest into their mathematical description also some growth rate functions that tells how the ribosomes can be related to effects on the growth rate. So for what concerns the models, we simply start from the most basal one that is typically used in synthetic biology, when we have the ribosomes that are splitted between the protein, the basal protein, the housekeeping one that allows the cell to survive, let's say, and the load that is what we are asking the cell to produce. Then we made a slightly more complex in which essentially ribosomes does not come only from ribosomal RNA, but also from some proteins that are integral part of the ribosomes. And the last one was found in some recent literature that it seems that exists a sort of negative feedback in the production of these proteins to maintain a certain level. So essentially, we try to develop these three models and apply to them different growth rate functions. So I would need like dozens of slides to explain all the combinatorial work we have done with the growth rate function. Just to summarize, essentially, we pick up a couple of type of relationship that are can be linear or Hill-like equation, and we never applied it directly to the total amount of ribosomes, actually more on, for example, ribosomes that are bounded to the basal protein. And for example, in the Hill, sometimes some models that there are some boundaries related to, for example, 30% of ribosomes are always required for the cell to survive and stuff like that. But at the end, we realized that the best models were the one, again, from relatively recent literature, that involves growth dependence with the active ribosomes, that are the sum of all the ribosomes that are already trying to translate mRNAs, so essentially bounded to mRNAs. And depending on the model, clearly this mRNA comes from the basal protein or the load that you are applying for the model 1, and also for the ribosomal protein pool of mRNAs. So essentially, how did we derive those models? So we know proteins, the basal proteins are transcribed, then there are the ribosomes that bound to this mRNA that can be bound and unbound, and then translated into our protein. And clearly, these ribosomes comes in the first assumption from the ribosomal RNA. So mirroring this structure to the load, we obtain the first model in which we have the ribosomes which are shared between the mRNA of the basal protein and the load. Clearly then, to pass to the model 2, we simply add another cassette for the ribosomal protein that now are also involved in the creation of the ribosomes. And clearly you see that the ribosomes now are shared between 3 actors. And the last of the model, it's simply with a negative feedback on the creation of the RNA by itself. So just a glimpse about how the modeling then worked. We simply started from drafting down the kinetic equation, then we derived the dynamic system. We also apply some mass conservation laws to see what was simplifiable and something on the fast kinetics and so far and so forth. And at the end, you always end up with a steady state structure in which, for example, for the complexes here, you have association and dissociation, the translation associated to that complex, a degradation, and also clearly also a dilution given to the growth itself. So very briefly about the results, clearly the first model that is not conceived to consider the growth rate inside was completely a mess. Indeed, you see that the more was the load, that was the blue line, actually the growth rate seems to increase at the beginning. And also the total amount of ribosomes starts immediately to go down. So moving back to the second model, now the situation is slightly better. Indeed, you see clearly now that we have an increase in the load that corresponds to actually a small decrease in the total amount of ribosomes, and then only after a certain threshold, the whole system starts to go down, both in growth rate and in amount of ribosomes and everything else. Even clearly, we still have a small increase in the growth rate and also a decrease in the ribosomes. Well, at the end, also adding the feedback, and now we have a system that acts as expected. Indeed, we have a certain region in which we have an increase of the load corresponding to a variation in the distribution of the available ribosomes among the other species, and only after the reaching of a certain transcription rate of the load, then we have a decrease both in the total amount of ribosome associated also with a decrease in the growth rate of the system. So to conclude, we did all this stuff just to catch out a couple of take-home messages, which are that if one wants to use this type of models to design novel genetic series for those type of application, you must for sure consider systems models in which you consider the active ribosomes for the growth rate models, and also the feedback on the production of the ribosomal protein. So what we are doing now, and trying to then publish the whole stuff, is try to demonstrate, validate this model also with the genetic circuits that are implemented in E. coli to see if we are able to get a decent data set to show that the model works. So I want to thank you all for the attention, clearly also the other co-authors and the other people with their work at the University of Padua, and also all the people that are involved in the 5 posters that are upstairs, and I hope you have enjoyed. Thank you.
Thank you, Massimo. Perfectly on time.
Good afternoon. Okay.
Yeah, so next speaker is Paola Facci from University of Roma Sapienza, and she will present her work on the long story of SWIM methodology from grapevine to personalized medicine. Oops, sorry.
Okay, thank you for the presentation. I'm going to present the SWIM, a network-based methodology that we recently developed in my lab, and which is able to identify a small pool of genes critically associated to a drastic change in cell phenotype. Okay. What is a network? You know, a network is a set of nodes and edges linking them, and a link occurs between 2 nodes if a sort of interaction occurs between them. And here you can see some famous type of different network. But why we need network? Because now it is well known that a disease is rarely the cause of a mutation in a single gene, but it is the consequence of the multiple interaction between different cellular components. And we need Why we need network? Networks are everywhere. We have molecular network, cell-cell communication network, nervous system is network, or social network, and network are powerful tool able to reduce complexities and are more efficient, more efficient than tables, and provide an intuitive visualization of your result and a common language between different disciplines. All of these, especially in biology. And now, recently, we developed Swimmer, that is the R version of Swim that was originally written in MATLAB, and it is freely available at this site. The phenotypic specific application of Swim are broad and include the identification of sweet genes in grapevine berry maturation, as well as in human cancer, and especially in glioblastoma. And this is some publication in plants or human cancer, and in the field of network medicine, that is an emerging field in the medical research. This is the step of the algorithm. I can't go into details of all the steps, but I can see that Swim allowed to reduce the complexity of several order of magnitude from the whole transcriptome, that is the input required from the tool, to more or less 100 genes that are the sweet genes. Now I'm going to present the interesting result we obtained by the application of Swim in glioblastoma. Glioblastoma is the most aggressive and frequent brain tumor, with a median survival time of only 12-15 months from diagnosis, and the mortality rate is extremely high, with 5 years survival rate achieved for only 5% of patients. And glioblastoma is resistant to the standard therapies like radio and chemotherapy, and now it is known that its aggressiveness is due to the presence of cancer stem-like cells that sustain tumor growth. And cancer stem-like cell share some typical characteristics with normal stem cell, like self-renewal and pluripotencies. And cancer stem-like cells are resistant to many conventional cancer therapies and cause relapse and metastasis. And thus, targeting cancer stem-like cells could pave the way for novel therapeutic strategies. We applied Swim to these dataset that was recently published on Cell, and in this paper the authors found a 4-core of transcription factor whose induction was sufficiently to reprogram the differentiated glioblastoma cells into cancer stem-like cells. Our aim was to identify genes, sweet genes, that could be related to the stem-like phenotype. And we use this further data set to validate our result. Sorry, okay. And this is the output of the first step of Swim, in which it identified differential expressive genes between two conditions, differentiated glioblastoma cells and cancer stem-like cells. And then it built the, no, excuse me, okay, no, I skipped, some slides are skipped, okay, no. Okay, don't worry. After computing differential expressive genes, Swim compute the correlation network, in which nodes are the transcripts and a link occurs between two nodes if they are highly correlated or highly anti-correlated. The strength of Swim is to take explicit into account the negative correlation. And then for each node in the network, SWIM compute the average Pearson correlation coefficient, that is the average of the Pearson correlation between the expression profile of a given node and the expression profile of each node in the network neighborhood. This parameter was used in the context of protein-protein interaction network in order to identify two classes of hubs, date hubs and party hubs, for positive value of this parameter. And no. Sorry for the problem. Okay, we were. Okay, this is the sketch of the input matrix. And okay, SWIM built the correlation network by using the Pearson correlation and by setting a threshold that should reflect the right balance between the number of network connected components and number of links. And so a reasonable threshold should be able to maintain the integrity of the network. And so this, with this threshold, we have a fully connected network. Increasing the threshold, we have that integrity of the network is destroyed. Okay, we were here. SWIM compute the average Pearson correlation coefficient for each node, and by computing this parameter, we found that the distribution of this parameter in the gene correlation network is trimodal. We recovered the date and party classification, but we found another peak for negative value of this parameter that correspond a new class of hubs that we called Fight Club hubs that are mainly anti-correlated with their network partners. And then SWIM used the K-means algorithm to find the communities in the network, and then it assigned a functional role to each node in the network by using the Guimara-Amaral approach that is based on the interaction of each node inter and intra clusters. In particular, they define two parameters: the participation coefficient that measure how node interact outside its cluster, and the within module degree that measure how nodes interact inside its cluster. And for instance, orange nodes are called module center or local hubs because they interact mainly in their community, and light blue nodes are peripheral nodes because they interact few in general, and green nodes are called connector because they interact both inside and outside their community. And okay, actually the situation is more complex, and we can define up to 7 functional roles. And this is the cartography map for the glioblastoma, in which each node is colored according to the APCC value, and we obtained what we call the heat cartography map. And remember, the negative value of the APCC correspond to Fight Club, and we can see a subset of Fight Club that fall in region 4 that are the network connectors. This is the, okay, and so the sweet genes are characterized by a high value of the participation coefficient, meaning that they mainly interact outside their cluster, they very low within module degree, meaning that they are not local hubs, and by a negative value of the APCC, meaning that they could act mainly as negative regulator. Okay, this is the result we obtained for glioblastoma in the 2 data set, and this heat map represents the expression value of sweet genes, and this heat map corresponds to the expression of their negative interactor in the network. In both data sets, we can see that there exists a group of sweet genes that are mainly upregulated in differentiated glioblastoma cells, suggesting that they could promote differentiation. They are involved in the focal adhesion pathways, and their activation strongly correlates with the repletion of the 4 core of transcription factors. Searching for common sweet genes between the 2 data sets, we found FOSL1 that is regulated in differentiated glioblastoma cells, and this upregulation correlates with the upregulation of important cell-cell communication genes in both datasets. But the most important result is that FOSL1 is downregulated in in cancer stem-like cells, and this downregulation strongly correlates with the activation of the 4 core of transcription factor in both datasets. And in order to identify a possible co-regulation of the 4 core of transcription factor, we search for a reached motif in the promoter region of the 4 core of transcription factor, and we found a consensus binding motif for FOSL1, and thus we suggested FOSL1 as a novel prognostic therapeutic biomarker which could induce differentiation of cancer stem-like cell by repressing the 4 core of transcription factor. And this result has been experimentally validated by a group of Policlinico Umberto I of Sapienza University of Rome, and are recently published on Scientific Report. I would like to thank all my collaborators, and thank you for your attention. And sorry for the problem.
Thank you, Paola. Okay, we can start the discussion now. Are there any questions from the audience? In the meanwhile, I would start with Paola. Have you integrated other types of omics data in your strategy? Actually, it works very well now, but it is possible to imagine an even more complex approach. For sure, it's a successful implementation of a systems biology approach where the reductionism is overcome, and we have the integration of many sources of data. So can you imagine the integration of other layers of information, other types of omics data?
Okay, Thank you for your question, and thank you for your consideration. And yes, of course, now we are collaborating with the Department of Medicine of the Harvard Medical School, and what we did is the new version of SWIM that is able also to deal with methylation data, and we are published the result of this integration between at the moment between two type of omics, transcriptomics and metylomics, and we are working on that.
It's a good development. And another more technical questions. I saw that you chose a basic correlation, a simple correlation. Have you explored other types of correlation, other types of way to estimate dependencies?
Okay. Thank you for also for this important question. Okay, the user can select Pearson or Sperman correlation. It depends from the data, and up to now I didn't implementing other type of nonlinear correlation like mutual information and so on. Since I'm driven by the idea that simpler is better, and up to now SWIM provide interesting results in a very different field, especially, for instance, in the grapevine berry maturation, in which we obtain a very, very important result. And so up to now, there is no necessity to implement further type of correlation, but for simplicity. But of course, this is another important task that could be implemented.
At the moment, the simpler, the better. It works very well. Okay, thank you.
Thank you.
I have a question. If one of the audience is ready to ask, I just skip, but meanwhile, I have a question for Massimo. So, in general, you addressed a very old question in biosystems biology with some quite updated tool. So that's good. In general, the questions are about how you expect to use that for practical applications, not only on the predicting what happens to the growth in a loaded condition, that is like a straightforward way to use your model. Not easy, but like you build it for that. But more on another long-lasting question in synthetic biology and biology in general, that is the dependency on the growth media, growth environment, because there are some parameters that could work, so could be tuned to understand what happens by changing it, and it would be great. That is the first point.
Okay, so thank you. It opened actually a pretty huge discussion if you want, but I'll try to be strict. So essentially I see, and actually the community is seeing different possibilities about this point. So essentially one could try to exploit this type, at least also my intention, was to exploit the things, for example, again, with a relation between different type of bacteria in the same culture, without going to touch the, let's say, chemostat effect or effects related to the nutrients. So just like adding an induction and through that tuning the growth rate. Clearly it can be done with many other possibilities, other techniques, but that's a possible one. Also because somehow, even if I don't have an example right now, you could also exploit it to tune at the same time the growth, but also the phenotype. For example, if you are expressing a load that gives a phenotype and you know that after a certain point you have a decrease both in growth and the expression of that one, you can have a double effect. Clearly, you need fine-tuning and so far and so forth. For what concern including in those system aspects which are related to the nutrients, so the media composition, clearly those models need to be expanded. And actually, this goes back to the first point of the fourth slide, I guess, that actually some models that take into account a relation between nutrients and growth, associating it with their absence, still exist. But those models are likely slightly less related to the metabolic load, or at least consider metabolic load given by the fact that it's growing slower because there are less nutrients and energy. So there are also some models can be added actually, why not, which are related exactly to the nutrients availability and so the energy. That's another approach, really.
And a more brief second point about a bit linked to that, there are a lot of parameters. Is there like a data analysis plan on how to estimate that from like experiments, the trivial ones, not trivial because they are easy, but the most obvious are loading the cells and check what happens, or using different type of strains, species, or
yeah, so
like this.
It's something in between, meaning that for sure, so for this type of simulation, we tried to make a big mess of the literature of the parameters that were estimated in more or less similar, let's say, experiments to the one that we are aiming to describe. And also finding that many parameters are really different, even if they expect to have the same behavior, let's say, very close experimental setup, or sometimes same experimental setup, paper claims to have different values of parameters, even order of magnitude, but that's not the point. In general, we will try to reduce, to change, let's say, one parameter per time. So for example, given the same strain, same growth media, trying to increase the load, and then maybe starting with, as you said, for example, a different strain, that's actually a good point, that starts, for example, because also the starting point is pretty important, from a certain growth rate that is defined, that you need to find a proxy to say how many ribosomes there are, see which are the differences, how they varies among different strains, and hopefully we can try to draw some conclusion also starting by the difference that comes from the starting point that might be different, applying the same type of load. That's the ideal approach at the beginning, at least.
Okay.
Thank you.
Please remain here. I have a question as well. Actually, ribosomal proteins are involved in high-level processes, not only canonical functions related to protein biosynthesis, but they are involved in high-level processes that impact on cell growth. They are pathways that can be p53 dependent or not dependent, and impact on cell growth and apoptosis triggering. Can you imagine, in perspective, a model that can include this kind of high-level information and description?
Okay, so if I'm correct, but you're also moving to like mammalian cells, like talking about p53. So that's a key aspect. All these things works for bacteria. Because, for example, it has been proved actually by people from University of Naples that, and IIT, I guess, in mammalian cells is not the translation the issue. When you have metabolic burden is due to the transcription resources. So is a different issue. For sure, also in bacteria you have other things that are related to the proteins, but at that point, I think we can start to discuss about which are the things that I need really to monitor, and we will have to understand if there is something that actually is worth to be modeled or not, or if we can just rely on that type of thing. For sure in mammalian, the mess is way bigger, so, but I don't have an answer about that. I deal with bacteria so far.
My point of view. Okay.
Thank you.
Thank you. One question for Deborah. I have a curiosity, I'd like to ask you some comments on specificity of receptors.
Okay, so the biosensors that we used were specific for a group of bioacids. So we don't want to substitute the current method of measurement of these bioacids. We rather measured a group of these biosensors are okay for our purpose, so because we want to control the concentration of classes of bioacids. So, so they are okay for this application.
Okay, okay. And the competition with mass spectrometry. What about it?
We didn't measure, we didn't compare the measurement of the concentration that we revealed with the biosensor, making a comparison with the mass spectrometry. So I don't know if we are more specific or no, but I think no. So I think that the mass spectrometry is more specific.
Yes, it's the ground truth at the moment. Thank you.
Thank you.
Any other questions from the audience? Okay, I think that that concludes this session. Thank you to the speakers for their interesting contributions, and thank you all for the participation.